In this video, we will discuss about the relationship of PDE4 and ER stress. More precisely, we will discuss the inhibition of PDE4 will prevent ER stress uh, that occurred due to this oxygen glucose deprivation condition. So what happens because of this oxygen glucose deprivation condition? There is a triggering of this ROS, or we can say reactive oxygen species, that will trigger the ER stress. So let us first understand what is the relationship between PDE4 and ER stress. So this PDE4 will interact with CAMP, and CAMP will interact with NRF2 and prevent them to go inside the nucleus. So when NRF2 is not go inside the nucleus, there will not be enough HO1 or hemooxygenase 1, enough hemooxygenase 1 production. So what will happen because of this is that the hemooxygenase will neutralize this ROS. So less availability of hemooxygenase 1 will leads to more amount of free ROS that will cause ER stress, and this ER stress will cause initiation of this signaling factor, that is PERK signaling factor and IRE1 alpha signaling factor, or we can say the increase in the amount of EIF2 alpha phosphorylated, and this triggering of CHOP, and finally what it will leads to, it will leads to this apoptosis. So what people have make a hypothesis that the inhibition of this PDE4 will free up this CAMP, so that in a way that CAMP will not be able to interact with NRF2, and so that NRF2 will move inside the nucleus, and it will leads to upregulation of this HO1. And this upregulation of oh, one thing that I need to clear is that when CAMP interact with PDE4, it will move towards 5 prime AMP, and when it will not interact with PDE4, it will move towards this NRF2, and NRF2 helps the NRF or helps the NRF2 to move inside the nucleus, so that it will upregulate the production of transcription of this hemooxygenase 1, that will be able to neutralize ROS. Thus, it will not trigger this ER stress. So, and this is one of the figure from the paper, the same paper. So that made that supports the point that increase in the amount of PDE4 will leads to higher ER stress. So it will use the opposing point to prove this theory. So what they did, they transfected the cells with empty vector and also the cells with PDE4B vector. So we can see that when the cells that are transfected with PDE4B in the western blot, the PDE4B production is more compared to the empty vector. And when they quantify this thing using semi-quantitative method, it is cleared that PDE4B is more in the cells that are transfected with the PDE4B vector compared to the empty vector. And then what they did is they check various proteins that are upregulated in the ER stress. So what they have find out that this P phosphorylated EIF2 alpha, that is this it is slightly increased, and this E and this, whereas the rest of the things are almost similar, or we can say this GRP78 is also slightly increased. So what they did in the next figure is that they quantify whether this increase is uh, quantitatively increased or not. So what they have identified is that there is an increase in the amount of GRP78, and also increase in the amount of phosphorylated EIF2 alpha and EIF2 alpha, phosphorylated EIF2 alpha, as we can see in the panel D and E. So So next what they did is this is when CDE are at normal condition, when there is upregulation of PDE4. So next they did is they introduced this condition, that is oxygen glucose deprivation condition, for 3 hours for cells, and then they measure then they do the experiment again. So what they have find out in that case is that in the western blot there is a change we can see in the phosphorylated EAF2 alpha and also in the GRP57. So they did the quantification analysis again to validate this thing, and they find out that there is upregulation of this GRP78, and it is more than what is upregulated in the normal condition. As we can see here, the value is around uh, 2.0, and here the value is around 1.5, the average value. And whereas for the empty vector, there is not much change that has been observed. And similarly for the P phosphorylated EIF2 alpha, the changes are more for the vectors that are that has more amount of PDE4B. Uh, there is more amount of PDE4 compared to a condition that is not observed this oxygen glucose deprivation. So this shows that increase in the amount of PDE4 will trigger more ER stress even in the normal condition. So in the from this figure they proved a relationship between PDE4 and ER stress. And if you need to understand this whole story in detail, you can go through the description that has been provided in the you can go through the reference that has been provided in the description. We will also discuss the other figure in the next videos. So that's all for this video. Thank you for your time.